I'm Balázs Kondoros. I'm a formulation scientist at Carbohyde, and I will talk about cyclodextrins in parental applications today. But first, I would like to start with the cyclodextrin science. What are cyclodextrins? I know that many of you know it, but I think today we have time, and it's better to start with the basics if we have this very uh, important topic like parental formulation. So after explaining what cyclodextrins are, we should know how to use them, how to make a complex of cyclodextrin and the API. There will be some slides about this one that okay, cyclodextrins are very nice molecules, they are very useful, but you also have to know how to use them to make an impact on the formulation. After that, we will check the main focus of cyclodextrins, like using them as excipients with small molecules. And most of the products are in small molecules still, but there are different approaches, like using them as excipients with biologics. We will see very nice examples and very nice scientific papers how to use them in this area. And after that, cyclodextrins and as active pharmaceutical ingredients. There are already some very nice examples and already one product on the market. So let's start. First, the general cyclodextrin background. So cyclodextrins are sugar molecules, ring-shaped sugar molecules, which can incorporate active pharmaceutical ingredients in their cavity. So the outer side of the cyclodextrin is hydrophilic, while the inner side is more lipophilic, and this very special property of the cyclodextrin can make feasible to complex the API, and by that the API's properties will be changed. So like the API has bad solubility, low stability, but this inclusion complex already can have higher solubility and higher stability. But what are these properties exactly? So the cyclodextrin in this way can decrease the taste, the smell, so the bitter taste of the API can cause problems, especially with children, and the cyclodextrin can solve this problem without using a large amount of sugar or any other ingredients. It also can decrease the interactions between drugs and drug excipients. The toxicity, irritation, and other side effects can be also decreased by these wonderful molecules. And you can increase the solubility. It is important because by increasing the solubility of an API, the bioavailability will be increased, so the efficiency of the API will be much better. The stability can be improved in the formulation, and also the wetting properties. And at the end, you will have a better formulation with higher bioavailability and maybe better stability. So they can be used in different ways. They can be very efficient and very useful in your formulations. There will be some questions. I don't remember exactly how many, but I would like you to answer these in the chat. I will wait a few minutes or seconds. So first question is, how many cyclodextrins are used in the pharma products? So maybe you already know these, so it's just a number. Please make a guess. I can give you a hint. It's 1 to 10, so between this range. I will check the chat box. We have an 8, 9, 9. Okay, I can say that it's very close. 5. Okay, thanks for being um, so productive and so contributing. So the answer is 9. seven for small molecules. These are the main focus of cyclodextrin, one for vaccine adjuvant and one as an active ingredient. So most of you were very close, or even very you knew the answer already. Okay, so let's see which are those cyclodextrins. You can see in this picture that and there are eight of them. You remember that I said nine, but in this slide we will talk about excipients, and I said that there is one API. So the nine excipients, alpha, beta, gamma cavities and their derivatives. You can see the orange bracket that shows which are the cyclodextrins that are used in parenteral formulations, and you can see that sulfolipo is like a dash line circled because it is not used in humans, only in vaccine adjuvants for veterinary use. But all in all, they are very popular in parenteral formulations, and all in all, more than 100 products on the market, and a huge portion of this is parenteral formulations, and they are used for solubilizing the products. So at the end, you will have a nice um, clear solution or a nice lyophilized cake. Also, in the formulation, it is very important sometimes to stabilize the API in the aqueous solution, so it will have a better stability. You can see here it's not the best or not so up to date this table, but it shows very well that most of the products with cyclodextrins are oral and parenteral, and that the focus of parenteral cyclodextrins are on hydroxypropylated and sulfobutylated beta cyclodextrins, and some are the hydroxypropylated gamma and one alpha cyclodextrin parenteral formulation. Why these two HP beta and sulfobutylated beta are the main focus? So the solubility of these derivatives are very high. It can reach 40%, which is a very uh, mm, huge advantage of these derivatives compared to other cyclodextrins, like beta has 1.5%, alpha has 20% maximum solubility in water. So by that, you can use a lot of cyclodextrin in the aqueous solution, and by that increase the solubility of the API even more. So it's not a big surprise that they are very popular and they are used often in this market. So the numbers may be not correct, so not up to date, but the focus and the trends are still the same. HP beta and sulfobutyl, they are dominating this market. So let's see the power of the cyclodextrins. In this first example, we will see how powerful solubility increasing effect it has. So alfaxalon, it's API that is used in 10 milligram per ml solution, but 10 milligram of alkoxalon can only be dissolved in 2 liters of pure water. But if you use a high amount of cyclodextrin, HP beta, in this example, you can decrease the amount below 1 milliliter, which is more than 10,000 fold solubility increasing, and someone already thought about it, and there is the product on the market. Alfaxan is the brand name. Besides solubility, cyclodextrin can increase permeation in certain circumstances. So usually we are saying solubility increasing effect of the cyclodextrin is basic, but in some cases permeation enhancement can be done by using cyclodextrins. For example, in this nasal product, Bakshimi, where they are using peptide formulation with beta-cyclodextrin, and the bioavailability increases 29 to 28% um, with the nasal powder formulation. It can be done because in the nasal cavity there is a high concentration of the cyclodextrin, and by that it can increase this permeation efficiency uh, with a high local cyclodextrin concentration. In an IV solution, it will dilute high mm, times, so it cannot increase the permeation only by increasing the solubility. And of course, stability increasement is important in the parenteral formulations where there are a lot of aqueous solutions. You dissolve your compound, and by that the risk of degradation will increase. So using cyclodextrin can improve this disadvantageous property of the API. In this example, you can see that it's an oral solution. So even in oral formulation, it can be important. In tablets and capsules, it's not that high importance, but in solutions like oral solutions and IV solutions or any other parenteral solution, it can be a huge difference. So we see beyond solubilization, we have other nice properties of cyclodextrin and others in tablets, for example, taste masking, content uniformity when we have low amount of API in the formulation, and also the fast dispersion is a very effective way of using cyclodextrin, but these are more oral focused properties. However, chemical stabilization and life cycle management are very nice examples for parenteral formulations as well. So what can be the issue with the stability? The light, the heat, the oxidation, the different pH, and the moisture can also degrade the APIs, and using cyclodextrins in a proper way. So if you incorporate the API in the cyclodextrin cavity, it can increase the stability by simply protecting the API from these effects. And there are very nice examples here for this. So how to make a complex? It's not a poll, it's just a question. And I will show a very short video uh, without sound. That in the first picture or first video, you can see that we are putting curcumin in the water. It's pure water. You can see that it's not wetting, it's not dissolving, its solubility is very low, so it's not a good, it has no good bioavailability. If you simply mix cyclodextrin, so just homogenize cyclodextrin with curcumin, you can see that in the second video that the wettability is already better, but the particles, you can see that it's still not dissolving. In the last one, we prepared the cyclodextrin complex, and you can see that in just seconds, this video is not, I don't know, faster than in reality. So just in a few seconds, it is dissolving, and the because of this, the bioavailability is expected to be much better of curcumin. Okay, so formulation technologies. I said that we prepare the formulation with curcumin, but which is the best one? So in this example, you can see that the physical mixture of the API and the cyclodextrin in 10 minutes it does not dissolve. Maybe it disperses, maybe some molecules are dissolved, but all in all, it's not the best. The lyophilized samples are dissolving in 10 minutes. In 5 minutes, you still see the particles, but with the electrospun nanofibers, you can see that already in 5 minutes it is dissolving completely. So, for example, with this technology, with electrospinning, you can see that even the preparation method of the cyclodextrin complex can make a huge difference, at least in the dissolution rate. So what is the process of complexation like this? First, we define the objective, the limitations, and the problem that should be solved with the cyclodextrin. Then we select the cyclodextrin, the technology to use, and if it's a solvent-based method, select the proper solvent for this process. Then, if we see that it does not work, we still have some modification options. For example, adjusting the pH, adding additional excipients. Sometimes these excipients has synergistic effects, so they can be very useful. And if we reach that solubility or stability level that we need, then we can evaluate it and maybe start over and select again a new type of formulation, and it can be a circle, and it can be a very long process. So how to evaluate it? Of course, it is the simplest one that you measure the properties of the API alone, the cyclodextrin alone, and the complex. In the first graph, you can see the DSC thermogram of an API, where you can see the melting point, but using cyclodextrin and making a complex can diminish the melting point, and by that you can see that the formulation has amorphous system. Also in the second figure, with the X-ray diffractograms, you can see that the crystalline peaks of the API can be decreased or even diminished, and with different type of other physical-chemical evaluation, you can characterize these products. So how to select the cyclodextrin? One of the most important and most used evaluation method is the phase solubility studies, where you use different type of cyclodextrin, different type of concentration of these cyclodextrin, and you check how the drug concentration increases with these cyclodextrin solutions. There are two main profiles for this phase solubility. One is B-type, where the complex has limited stability. For example, you can see I is like the solubility is not increasing or increasing with BS for a certain amount, but after that it's like a plateau. With AL, which is the most suitable for formulation, it's increasing with the cyclodextrin concentration, and with the cyclodextrin derivatives, usually we see this one. So if the incorporation of the compound is feasible, then we will have an AL type of profile. Okay, so we selected the cyclodextrin, then we have to select the preparation method, because we need this the complex. How to select it? Of course, you have to know what is the final formulation. It will be a solid or a liquid. What are the API's properties? For example, if you have a heat-sensitive API, you will not use spray drying, but rather freeze drying. So these steps can be very tricky, but there are a lot of equipment and a lot of different technologies that can be used with cyclodextrins as with other excipients. So this slide is quite heavy, but it can be a very nice guideline if you want to uh, choose one method to use with your API, and you need to choose between freeze drying, co-grinding, spray drying. So maybe if your compound is mm, uh, moisture sensitive, you can use co-grinding where you don't need to add any solvent. You just grind it in a dry method. It is also eco-friendly. So even in this grinding process, the complex can be formed. But there are a lot of different type of methods to use. As I said, I will share this so you can check it later. It is very heavy. I won't share every information that is on the slide, or you can take a screenshot. Okay, so as I said, cyclodextrins or using cyclodextrins are more focused on the small molecules, uh, and I will talk about it now. But first, a question, what do you think, what is currently the highest approved daily dose of a cyclodextrin? I will wait a few seconds, and I can give you a hint that it's a parenteral formulation, and it's more than 1 gram. Okay, someone wrote 100 milligram. No, 1000 milligram, sorry. 2000 milligram, 1.5 grams. Okay, these are still low, I would say. But if there are no more questions or tips, so it's 16 grams and it is used in a parenteral formulation. Sulfobutyl rated cyclodextrin can be dissolved in a 40% range. So this 16 grams can be administered by this product. My slide is okay. So we saw that this high amount of cyclodextrin is used, of course, because it's needed, because high amount of cyclodextrin is needed for the APIs to be dissolved, but you can decrease this cyclodextrin concentration by using a third component, for example, hydroxy acids, amino acids, metal ions, or water-soluble polymers. Sometimes these additional excipient has solubility increasing effect. Also, the cyclodextrin have a solubility increasing effect, but using them together, they can have a synergistic effect, and the amount of these excipients can be drastically decreased by this ternary, so-called ternary cyclodextrin compositions. So one more very nice example that 10 milligram of domperidone can be dissolved in 2 liters of water uh, in 62 milliliters of cyclodextrin solution, in 12 milliliters of hydroxy acid solution. But if you combine this cyclodextrin with the hydroxy acid, it can be dissolved in less than 1 milliliter of water, which is a huge increase in solubility. Again, a formulation containing API where they use two different type of solubility increasing mechanism. First is the preparation of the salt form. You can see that they used free base, tosylate, hydrochloride, and mesylate. And in these, you can see that the solubility in water was the best with mesylate salt. Um, Around 1 milligram per milliliter. Uh, and after they chose two cyclodextrins, of course, HPBeta and SBCD, and you can see that between those with the salts, sulfobutyl in 40% with mesylate salt was the best, 44 milligram per milliliter, and if you compare it to the free base in water without cyclodextrin, it's 0.3 microgram per milliliter, so it's again thousand-fold or 10,000-fold increasement in solubility. And one of my favorite example, it's the life cycle management of the of the different products. It is a very nice example because it has a scientific impact and a strategic impact as well. So replacing these excipients, for example, polysorbates, different type of mm, solvents or mm, polymers, you can increase the safety and the efficiency of the product, and also it has a strategic impact that maximizing the value of the API and using it in different unmet medical needs or innovative ways to have a new product. The example is with melphalan in the old formulation or replacing the old excipient. I use this quote to quote because cyclodextrins are not new, they are just as old as others, but replacing them with the previous one, it can have a huge impact. So formulation one includes one lyophilized powder with the hydrochloride salt and povidone, uh, and also a not standard solution of propylene glycol, ethanol, and of course water. So it's all needed, the hydrochloric salt, the different type of diluents, the solvents, to just have a final clear solution of the product. And also it needs a standard diluent, which is very problematic in the clinics to use. However, in the new formulation, again new, you have a lyophilized powder of the free base API, so not even the hydrochloric salt is needed. You just use the basic free base API with sulfobutyl cyclodextrin, and you can use a standard solution to reconstitute in the clinics. Of course, again, you have to use a large amount of cyclodextrin. In this case, maybe you don't see the slide, but it's 2.7 grams per 10 milliliters of cyclodextrin. But you could change the different type of other excipients with this solution. So yes, injections and infusions are on the market. Mostly sulfobutyl and HP beta are used in certain cases, alpha cyclodextrin or HP gamma cyclodextrins. And the final product is a solution or a powder, a lyophilized powder, which can be reconstituted easily for administration. However, there are some intramuscular and subcutaneous products. There is one subcutaneous product, I don't remember the name, but it's a very important thing to note that because of using high amount of cyclodextrin, the subcutaneous formulations are limited because they are low volume. So you can't dissolve grams of cyclodextrin in a low volume, but there is one cyclodextrin product, and for sure it's with HP beta cyclodextrin because sulfobutylated cyclodextrin is a sodium salt. So if you use this sodium salt, you will have a high osmolality, and because of that, you will have tissue irritation. So in subcutaneous, for sure, I would go with the HP beta. They can be combined with different type of formulation approaches. For example, with liposomes, uh, it's just one representative example that you incorporate the API in the cyclodextrin, and it is now in an aqueous solution. And by incorporating this aqueous solution in the middle of a liposome, you will have a nice structure with probably with a synergistic effect, and by that more effective way to use this API. Uh, to my knowledge, there are no marketed product with this combination, but it's feasible. I'm sure that it increases the efficiency of the formulation, but a little bit more complicated than just using liposomes or just using cyclodextrin. Maybe if you know them, if there is any marketed product, please tell me in the chat. Okay, so I said that cyclodextrins are, or cyclodextrin field are still focusing on small molecules, and it was in the past, it's still in the future, uh, and but it's broadening. So focus is still on the small molecules, but broadening to biologics and APIs. So what about proteins? Proteins have certain uh, disadvantages, like they can aggregate. Sometimes you have to use them in high concentration, so the viscosity increases as well. They can be adsorbed to the wall of the vial or the mm, glassary. The solubility can be low sometimes. Usually it's not a huge problem, but sometimes solubility is not good, and also the degradation of the the proteins are a huge problem. But cyclodextrin can solve these problems. So there are examples for this, for all of these problems. So decreasing the viscosity, decreasing the absorption, and so on. But how? So what is the mechanism of protein stabilization? Because I said that cyclodextrin works like incorporating the API in the cavity, but proteins are just large. They cannot fit any cyclodextrin cavity. So how it's working? It's not sure yet. There are a lot of scientific papers and a lot of mm, evident, but we don't know yet. There are some mechanisms like the cyclodextrin can interact with the part of um, the proteins, as you can see in the picture, or the other mechanism of actions like with the polymers, they can interact with the surface of the proteins, so they can increase this solubility, decrease the adhesion to the wall. Uh, yes, still cyclodextrin interacting with proteins for sure. There are no parenteral products on the market yet, but there are a lot of information we already have, and they have a potential because they have similar effects like polysorbates that are used in the uh, monoclonal antibody products, for example, and again, they can be changed for cyclodextrins. Cyclodextrins are safer to use than surfactants like polysorbate. There are no degradation, which can cause, in case of polysorbates, peroxidate fatty acid formation, and cyclodextrin can have certain advantages. As you can see in this case study done by Rocket, where they are using adalimumab as a monoclonal antibody and HP beta and polysorbate 80. You can see that Kleptos, which is the cyclodextrin, is used in 50 millimolars, which is around 7% cyclodextrin, while polysorbate is used in 0.1%. So in this, you can see the particle concentration, and in this case, the lower the number, the better. So you can see that without using any type of excipient, it has a high value, which is not suitable for the formulation. But if you are using the cyclodextrin or the polysorbate, you have somewhat similar effect. But if you are using polysorbate and cyclodextrin, it has a synergistic effect, and you can see that after 2 hours, the value is still very low when you are using both excipients. This company, Rocket, has already a product on the market, cyclodextrin product, which is focusing on monoclonal antibodies. It's called Kleptos Biopharma, and they are focusing on monoclonal antibody formulations. However, there are still no pharmaceutical product on the market with this approach. But there are some with smaller molecules, with peptides. For example, tetrapeptide carfilzomib is used in a clear IV solution with sulfobutyl, and Baqsimi that I already mentioned. It's a glucagon-containing product, which is a protein hormone. We are using beta cyclodextrin, and the final formulation is a nasal powder. And by that, in the first example, Kyprolis is using cyclodextrin for solubility increasing, while Baqsimi using cyclodextrin for permeation enhancing. Just few words about sulfolipocyclodextrin, because it's very rare that they are using this. It's a special cyclodextrin derivative that is used as a vaccine adjuvant in a veterinary injection by Pfizer. But not just this special cyclodextrin is used as adjuvant, also HP beta is used. You can see here that Japanese company Daiichi Sankyo, which is a large big pharma company, is using HP beta for intranasal influenza vaccine. So the cyclodextrin can increase the T cell related immune response and reduce the allergic risk to the vaccine, which is again a very nice, very advantageous property. They can be vaccine adjuvant to increase the effect of the vaccine, but they can be stabilizers as well to increase the chemical and physical stability of the product. For example, in this example with Sanofi, with the poliovirus vaccine, where they are using also thiamerosal and HP gamma cyclodextrin. Other than that, they are also cryoprotectants. So in some vaccines, for example, in Janssen COVID vaccine, they are using HP beta for cryoprotectant, not for other reasons, but just to have a nice final freeze-dried formulation. It is already on the market, as you may know, but there are some patented formulations, for example, methylated and hydroxypropylated cyclodextrin with T cells. They could have a nice final cell lyophilized sample at the end. Also, cryopreservation is possible with cyclodextrin, for example, with fertilization in veterinary use. Okay, so last topic is cyclodextrins as APIs. You can see here few, or not that few, a lot of companies that are using cyclodextrin in products, but not as excipients, but APIs, active pharmaceutical ingredients. A lot of uh, them are already on the market, but some of them are just in preclinical phase, and we are the part of this group, so we also focusing on the API side of cyclodextrin, not just the excipient side. So there will be some AI-generated pictures here and some puns. I apologize for this. I didn't do it. My uh, colleague Tamás Sohajda did these. So to demonstrate how they are working, there is two mechanism of using cyclodextrin as API. One of it is a scavenger type, as we saw before, as with the excipient focus, that they can be incorporate different type of molecules. So of course we can use it in the body to scavenge some API or some molecules and eliminate it from the body. The other part is the cholesterol modulator mechanism, where they are using the effect of cyclodextrin immobilizing the cholesterol. So it incorporates cholesterol and it works like that. So again, a pun that it's Captain Scavenger, Bridion. It was the first marketed product as a cyclodextrin API. It is used after after surgeries where they are using glucuronium, and of course you need the glucuronium, but when you don't need the effect anymore, you can inject Bridion. It's complexing the API and eliminates from the body, and it can be done by modifying the cyclodextrin, so not just the complexation effect, but also ionic effect of the negative charged cyclodextrin, and the positively charged API play a role in this effect, and this complex is very strong, so it will not not dissociate, and will be eliminated from the body. As you can see in the picture, in the slide, that it was approved in 2008 in the EU, and just 7 years later in the US, because first in the US the FDA declined this product, but 7 years later it was introduced to the market, and at the end it was a blockbuster drug. In certain areas of the world, the patent already expired, so there are some generics of the original Bridion, which was introduced by MSD. Same group, the scavenger type of molecules. Besides sugar madex, there are different designed molecules for different APIs, so you have to find the perfect fit to the API, and by chemically modifying the cyclodextrin it can be done. So there are some sugar madex generics, as I said, and for example with Adam Gamadex, it is the same purpose. It's just a different molecule, but does the same. But there are different aims. For example, to fentanyl overdose, they are using another type of cyclodextrin, and so on. The other part was the cholesterol modulators, and this story starts with H3BCD. In the first literature data, they used a special type of API with cyclodextrin. The cyclodextrin was an excipient, but with the mice studies, they also used solutions without API, so only cyclodextrin was inside the formulation, and they saw that with cyclodextrin and without API it works better. So the cyclodextrin must have some kind of effect, and by that they started to investigate Niemann-Pick C disease, which is a cholesterol hemostasis mm, related disease. And but not just this Niemann-Pick C is in this cholesterol hemostasis disease category. There are different types like Alzheimer, Parkinson, lymphedema, dry eye disease, and for the same type of action, you can use hydroxypropyl beta cyclodextrin as well. So it's now a league of the cholesterol-related issues. So what's beyond HP beta, what's beyond cholesterol modulation? We are focusing now in this, but there are new innovations always with cyclodextrin, even though we know them for decades. There is always innovation in this field. So we are closing this session with the take-home message that cyclodextrin can be used and are used in the pharma products. Safety is not a problem if you know how to use them. Mainly they are still used in solubilization and stabilization, and they can be used for replacing other excipients or preparing new type of products. And there are novel applications, API adjuvants, so really the cyclodextrins can be used in different ways, a lot of different application area. So if you have any questions, here is our contacts, Tamás Sohajda, my colleague, and of course you can reach out for any of us um, and contact us in LinkedIn, in the carbohyde.com, in different ways. Okay, so thank you for your attention. This is the end of the presentation. I hope you had some new information and you learned some new things, and I hope we will have a nice conversation here. I am checking the chat. Okay, so one question was how cyclodextrins are different from phospholipids in case of increasing bioavailability. So cyclodextrins and phospholipids can increase both the bioavailability increasement. In most cases, cyclodextrin only increase the solubility, and by increasing the solubility, more APIs will be ready next to the cells to be absorbed. So this is the mechanism of action. With phospholipids, there are more complex mechanisms, and the permeation is increased as well with phospholipids. However, preparing phospholipid formulations are more expensive. There are more issues related. So I would say both cyclodextrins and phospholipid can be used and should be used in the market, but they have still the different approaches. In case of if the question was how high is the bioavailability increasement effect, I would say that depends case by case. Sometimes cyclodextrins are better, sometimes our phospholipid based formulations are better. So in a formulation development, you should try both. Is this product, the 16-gram cyclodextrin, approved in a regulated market? Yes, it is approved by FDA. I'm sorry, but I don't remember the name. But it is in a regulated market, and what was the difference between cyclodextrin mixture and cyclodextrin complex of curcumin? So if you remember the video, the question is for that. With the mixture, we just homogenized it in a mortar, so not grinding, but just homogenizing cyclodextrin and curcumin. With the complex, we used freeze drying or spray drying, I don't remember, but one of them. And by dissolving cyclodextrin and curcumin and then making this complex by this method increased at least the solubility rate, dissolution rate, but I can tell that we checked the solubility as well, and the solubility was increased as well compared to the mixture. How does cyclodextrin increase dissolution rate and GI bioavailability? So the cyclodextrin increases the dissolution rate by incorporating the API, and because the API is not is inside the cavity, and the cyclodextrin outer surface is well soluble, so it gets molecularly dispersed, the whole complex, and by that it will increase a lot compared to the very low solubility API, and by that you have this molecularly dispersed solution, and the cyclodextrin and the API is dissociating, so it's a dynamic equilibrium. In 1 second, it's goes in and goes out of the cavity 1,000 times or even more. So when it's leaving the cavity, it can be absorbed to the GI tract. Are the industry utilizing cyclodextrin for increasing bioavailability of GLP-1 inhibitors? Yes, there are patents, there are different type of approaches to use cyclodextrins, and they have benefits, but there is still not any products on the market with GLP-1 inhibitors yet. But I would expect in a few years that we will see the first preclinical or clinical studies. There are preclinical studies already. What are the other competitor molecules like cyclodextrins used in pharma industry? So in this question, I think you are asking if there are any molecules that can be used instead of cyclodextrin. Of course, you can use solvents, but they have different problems. So irritation, regulatory problems, also polymers you can use, but maybe they have higher viscosity at the end. Polysorbates, I mentioned that industry really don't like polysorbates nowadays, so they want to change those. So yes, there are a lot of different type of excipients that can be used for increasing the solubility, increasing the stability, and so on. But there are just a few that can do all in one, and cyclodextrins are just great in some cases. According to the pharmacopoeias, European and the US pharmacopoeia, alpha, HP beta, sulfobutyl are approved in products to be administered parenterally. However, the remaining ones, is their use prohibited in parenteral drugs? Are there documented safety issues? Yes, so this is for a reason. They are on the market for decades, so as I said, alpha is the only only of the three parent cyclodextrin that is used because beta cyclodextrin has low solubility, around 1.5% in water, and when you are administering it in a large volume, so for example 1 gram is needed sometimes, you need a large volume first, and after it can precipitate in the body, which is which can cause problems. So nephrotoxicity is documented as a safety issue with beta cyclodextrin. And yes, this is the main reason beta is not used. Yes, HP beta, sulfobutyl, and alpha are used. And also HP gamma. I hope I answered this question, but if you have more, please contact me or write another message. Should we worry that the complex with cyclodextrin may dissociate in human circulation system? We should not worry about it because this is the aim. So if the complex is so strong that it does not release the API, then it will not be um, absorbed. So we aim for this to dissociate and by that to be uh, absorbed to the body. Where is the best place to find information on viscosity and percentage of cyclodextrin? Uh, there are some articles for sure. I have collected them in one place. If you write a message for me on info at carbohyde.com or formulation at carbohyde.com, I can send you this data. What is the maximum allowable concentration of HP beta and sulfobutyl in drug products for parenteral administration? I cannot tell you this now, I don't remember, but you can find it for the FDA. There is an inactive ingredient list, and you can see which excipient can be used in which administration route and what is the maximum allowed concentration. But it's quite high, as I said, that in some cases it's more grams daily dose. What are the possible risks for human health when cyclodextrin concentration is too high? One of the main issue is nephrotoxicity, as I said. Also, ototoxicity was recorded. So they are really used in gram scale. If they are accumulating in one part of the body, they are sugar molecules. They can cause a lot of problems, like uh, in the ears or in the nephrons. So this is the main issue. They are they cannot be absorbed. So the bioavailability of most of the cyclodextrins are below 1%. It is not an issue. The accumulation of the cyclodextrin in one specific part of the body can be an issue. What kind of molecules are more compatible with cyclodextrin molecules with low log P or high log P? So the more lipophilic the drug, the more it goes inside the cavity because it's really don't like the aqueous solution, but likes the cavity of the cyclodextrin. So I would say this is a rule that if it's very low solubility, it will like the cyclodextrin cavity, but we can say that if something is more lipophilic, then it will have a very nice complex. There are other factors that influence this cyclodextrin complexation, not just the log P value. We are already 5 minutes more than uh, I said, and I think there are no more questions. I really like this occasion, and I hope that you as well. So thank you for joining. Thank you for the questions. So meet you in the next one. Bye bye.